Have you ever pondered the significance of identifying fixed oils in the pharmaceutical industry? Fixed oils, sometimes referred to as base oils, are the unsung heroes of the pharmaceutical world. They are essential components in a myriad of medications, and their roles are as diverse as they are crucial. Imagine a world without fixed oils. Medications would lose their stability, and the delivery of active ingredients would be compromised. This is because fixed oils often act as carriers for medications, helping to transport drugs safely and efficiently to their intended targets within the body. But that's not all. Fixed oils also contribute to the formulation of drugs. They can help to modulate the release of active ingredients, ensuring that medications act at the right pace and at the right time. Without them, the finely tuned balance of drug action could be thrown off kilter. Perhaps even more critically, fixed oils play a key role in maintaining the stability of pharmaceutical products. They can help to prevent the degradation of drugs, extending their shelf life and ensuring they remain effective even after extended periods of storage. However, not all fixed oils are created equal. Different oils have different properties, and choosing the right oil for a particular medication can be a complex task. This is where the identification of fixed oils comes into play. By accurately identifying the fixed oils used in a pharmaceutical product, manufacturers can ensure that their medications are safe, effective, and stable. They can guarantee that their drugs will work as intended without any unwanted surprises. The process of identifying fixed oils is not always straightforward. It requires a keen eye, a steady hand, and a deep understanding of the unique characteristics of different oils. But the rewards are well worth the effort. By identifying the right oil for the job, manufacturers can create medications that are not only effective, but also safe and reliable. Indeed, the identification of fixed oils plays a crucial role in the creation of safe and effective medications. So how do we accurately identify these fixed oils? Enter Thin-Layer Chromatography, or TLC. This analytical technique uses a thin layer of absorbent material such as silica gel or cellulose, fixed onto a flat, inert substrate. A liquid sample is applied to this plate, and a solvent, or mixture of solvents, carries the sample up the plate via capillary action. Now, where does its magic lie? It's in the separation. The different components in the sample move at different rates, separating into distinct spots. These spots can then be analyzed to identify the components, like our fixed oils. In the pharmaceutical industry, TLC is a star player. It's used not just for identifying fixed oils, but also for monitoring chemical reactions, identifying compounds present in a given substance, and even in forensic science to detect drug use. With its simplicity, affordability, and accuracy, TLC has become an essential tool in the pharmaceutical industry. The first step in thin-layer chromatography is sample preparation. That's right, before we dive into the actual process of chromatography, we need to ensure our sample is ready to go. So, how do we prepare the sample? It's not as tricky as you may think. The key is to have a clean, accurate sample to begin with. Think of it like a chef, ensuring their ingredients are fresh and clean before they start cooking. In the same way, we need to ensure our sample is free from impurities and is of the right concentration. To prepare the sample, we'll need to dissolve it in a suitable solvent. This could be anything from water to alcohol, depending on the nature of the sample. Remember, the solvent should be selected carefully, as it needs to dissolve the sample completely without causing any chemical reactions. It's a bit like picking the right wine to go with your meal. The solvent and the sample should complement each other perfectly. Once the sample is dissolved in the solvent, we'll need to filter it. This is to remove any undissolved particles that might interfere with the chromatography process. It's like sifting flour before baking. You don't want any lumps ruining the smoothness of your cake, do you? Similarly, we want our sample to be as pure and smooth as possible. Now, this step might sound simple, but it's crucial to get it right. The accuracy of thin-layer chromatography depends heavily on the sample preparation. A small error here can lead to inaccurate results later. So, Take your time and do it right. After the sample is prepared, it's time for the next step, applying the sample to the plate. But that's a topic for another time. For now, let's remember that sample preparation is not just a step in the process, it's the foundation upon which the whole process is built. Proper sample preparation is the cornerstone of accurate, thin-layer chromatography. So, remember to give it the attention it deserves. After all, a strong foundation is essential for a sturdy structure. Let's keep that in mind as we move forward in our exploration of thin-layer chromatography. Once the sample is prepared, the next step is applying it to the TLC plate. Now this is where the magic really starts to happen in the world of thin-layer chromatography, or TLC, as it's often affectionately called. The TLC plate, for those who may be new to this process, is a sheet of glass, plastic, or aluminum foil coated with a thin layer of a solid, adsorbent material, usually silica gel. This is known as the stationary phase. So, let's dive into the application process. It's all about precision. A small amount of the prepared sample is applied to the plate with a micropipette. We're talking infinitesimal amounts here, folks. It's crucial to apply the sample as a tiny spot, about 1 millimeter in diameter, onto the plate. And this spot needs to be about 1.5 to 2 centimeters from the bottom of the plate. Why is this so important? Well, it's because the sample needs to be above the level of the developing solvent. If it's not, it could dissolve and spread out in the solvent, which would interfere with the separation process. We certainly don't want that. Now let's shift gears a bit and talk about the stationary phase. This is where our sample will interact, and this interaction is what separates the different components of the sample. In TLC, the stationary phase is polar, meaning it has a slight electrical charge. This charge is what attracts the components of the sample. Each component has a different level of attraction to the stationary phase, and this difference in attraction levels is what separates the components as they move up the plate. So as you can see, the application of the sample to the TLC plate isn't just a step in the process. It's a critical moment that sets the stage for the entire separation process. It's a delicate dance of chemistry and physics that, when done correctly, provides us with valuable information about the fixed oils in our sample. The accurate application of the sample to the TLC plate is critical to the success of the process. Remember, in the world of thin-layer chromatography, every tiny detail counts. With the sample applied, we now move on to development and visualization. This is where the magic of thin-layer chromatography, or TLC, truly comes to life. The development stage begins by immersing the lower edge of the TLC plate into the solvent. Here, it's crucial that the solvent level is below the applied sample spot to prevent it from dissolving directly into the solvent. As the solvent rises up the plate by capillary action, it carries along the substances in the sample. The different substances move at different rates, separating along the way based on their interaction with the stationary phase of the TLC plate. Once the solvent has almost reached the top of the plate, we remove it from the developing chamber and allow it to dry. This drying process solidifies the positions of the various components on the TLC plate, translating the hidden details of our sample into a visible form. Next, we move on to the visualization of these separated components. Now, this is where things get really interesting. Depending on the nature of the substances, there are a couple of methods used to visualize the results. One common method is to examine the TLC plate under ultraviolet light. Many organic compounds absorb UV light and re-emit it as visible light, allowing us to see their positions on the TLC plate. In cases where the substances do not fluoresce under UV light, we can use chemical reagents that react with the substances to produce visible colors. It's like creating a beautiful, intricate map, where each spot or streak represents a different component of our sample. By comparing these spots with reference substances, we can identify the various components in our sample. Through development and visualization, the individual components of our sample become visible, allowing for their identification. This is the power of TLC. It deciphers the complex language of chemical substances, translating it into something we can understand and analyze. And with this knowledge, we can ensure the quality, safety, and efficacy of our pharmaceutical products. So let's bring our discussion on thin-layer chromatography to a close. We started our journey understanding the integral role that fixed oils play in the pharmaceutical industry, from serving as carriers of active ingredients to their use in various formulations. We then delved into the world of thin-layer chromatography, a powerful technique that helps us identify these fixed oils. We walked through the detailed steps of a TLC procedure, beginning with the careful preparation of the sample, ensuring it's appropriately diluted and ready for analysis. We then moved to the application of this sample onto the TLC plate, taking care to apply it as a small spot at the base. Our journey didn't stop there. We then developed the plate in a suitable solvent and visualized the separated components under ultraviolet light or by using a suitable revealing agent. And that, dear viewers, is the fascinating process of thin-layer chromatography, a vital tool in the identification of fixed oils in the pharmaceutical industry.